classification.
Loose fibrous
connective tissue taken from subcutaneous layer of the skin and also stain. You also need to describe the stain. Iron hematoxylin. Yes, if you see dark blue color, this is iron hematoxylin. Now your brief description, which includes only major morphological properties. So how can I identify this image, classify this image as loose fibrous connective tissue? First of all, I can see two types of fibers: collagen fibers and elastic fibers. Pale stained thicker collagen fibers and sharp, darkly stained, very thin, elastic fibers. Then, what else?
Fibroblasts. Fibroblasts.
Yes, come in. We have just started. Then, by importance. Yes, the next feature which is clearly visible. Predominance of ground substance. Yes, ground substance.
Extracellular matrix.
Yes, which is naturally extracellular matrix, which is a dominant component. In this case, it remains unstained. So I can see many empty spaces, but I understand these are not empty spaces.
Ground
this is ground substance. And this is the most specific property of loose connective tissue. Yes, you remember predominance of ground substance over cells, over fibers. Then loose arrangement, so irregular arrangement of all fibers. And finally, not abundant, a cellular compartment represented by fibroblasts. Yes, fibroblasts. Also loosely organized. Without specific direction, that is all. Yes, that is all that you can see by morphology. In low magnification, you may say some other cells may be present. Yes, it is diverse in composition, but the major type of cells, fibroblasts, also cells with irregular shape, irregularly branched cytoplasm, this will be enough, yes, for you. We move further. In contrast, what is this? Another type of fibrous connective tissue, now dense regular.
Dense connective, dense regular.
Yes, full classification. This is dense regular fibrous connective tissue. On the example of a tendon stained by hematoxylin eosin, how can I identify? What helps me to classify this slide? First of all, I can see now highly organized. Yes, highly organized, arranged in parallel, strictly in parallel, long collagen fibers, tightly packed, with no spaces between them, arranged as bundles of the first order, separated by what? By very highly elongated nuclei of fibrocytes, yes, or tendinocytes. Yes, tendon cells, tendinocytes. Bundles of the first order, and what should be noticed? Collagen fibers, you remember, show oxyphilic properties, yes? Stained by eosin. Yes, I can see bright oxyphilic, highly arranged, in parallel, strictly in parallel. Bundles of collagen fibers. Which are arranged into higher order of bundles of the second order, yes, separated by interlayers of loose connective tissue called endotendinum. That is all. You see, I take only the properties that are visible, with no details. We need no details for identification. Moving further. Another case, 2 slides, you will see. This is low magnification, yes, low magnification image, and the same but in high magnification, 2 images. How? Let's try to identify this image.
Specialized connective.
Yes, specialized. We need to explain. So this is specialized reticular. Yes, reticular specialized connective tissue taken from
Lymph node.
yes, the
Lymph
stroma
node.
of the lymph node. What may be an explanation? How can you confirm your choice? Here I can see a spongy structure, yes, sponge-like structure,
Spongiocytes.
porous. Stroma is arranged as a meshwork, yes, a meshwork or a network. This is due to specific reticular cells, right? Reticular fibroblast-like reticular cells with many processes. If we look at the background, these cells are oxyphilic. Yes, oxyphilic. They make up an spongy architecture. Yes, reticular architecture of a tissue, which function as a scaffold for another cell type. I can see also many small rounded nuclei. These are lymphocytes. Yes, lymphocytes. So reticular cells, branched cells with processes that make up a sponge-like architecture. In between these processes, like in pockets, I can see lymphocytes. That is all. Yes, quite enough for this image, with no details. Then what is this? Easily recognizable cartilage. Yes, hyaline cartilage. This is total view, but you need to provide the origin from where it is taken. This is the cross section through the ventral ends of ribs with articulation with the sternum. In high magnification, yes, I will show you in low and high magnification. In high magnification, what is the most remarkable, the most recognizable property? Isogenous groups, right? If I see large rounded cells, with whitish cytoplasm, yes, whitish cytoplasm. These are chondrocytes, you say, chondrocytes. If they are organized in groups by how many? 3, 4, yes, generally 3, 4, 5. Each group occupies its own lacuna, yes, lacuna within the matrix. And this lacuna is immediately surrounded by dark violet, yes, dark basophilic matrix, known as territorial matrix. Obviously, this is a hyaline cartilage. Between territorial matrix, we can see less basophilic, transparent. Interterritorial matrix, transparent. Also a feature of hyaline cartilage, transparency. Yes. Then this will be enough, but you can complete, yes, your explanation. On the periphery, I can see I can observe perichondrium, and I can clearly see the two zones: young cartilage, yes, and mature cartilage. Young cartilage contains only single
chondrocyte.
separated chondrocytes, not arranged as groups. This will be enough, yes, for the description. You can notice that I am based on the legends, yes, to your drawings, which contains definitions that are visible. Not all the material, all that we should remember about a given tissue type, only what is visible. So you can, this is just a suggestion, a recommendation. You can combine these definitions from legends to construct a short description. Yes, you need to complete by probably description of the shape, yeah, the shape, the staining properties, but you can use these basic definitions from figure captions. This is again loose connective tissue. Fibrous. Subcutaneously, you can see the different variants of slice, different appearance probably, but anyway, you say, yes, anyway, you can start with collagen fibers, elastic fibers, ground substance, and finally fibroblasts. The same, yes? loose fibrous connective tissue. A little bit different appearance, but the same. And once again, loose fibrous connective tissue. What is this?
Adipose. Bone cell.
Bone. Yes, this is, but full title. This this is secondary lamella
Secondary
bone
lamella.
taken from a diaphysis, yes, of long tubular bone, decalcified, yes, decalcified, stained by specific Schmorl stain. What is the most identifiable structure? Yes, yes. I can classify. I can
Haversian canal
recognize it
system.
by a layer of concentric Haversian system or other osteons, and you say each osteon as a structural unit consists of several concentric lamella, yes, inserted into one another with a central canal, and I can also see lacunae, yes, many lacunae for osteocytes, and irradiating canaliculi. Yes, we remember each lacuna sends canaliculi containing processes of osteocytes. In principle, this is enough. Yes, nothing more is visible. Another slide, another variant of the same section. Yes, the same slide. Once again, Haversian system, central canals containing blood vessels, you can say, and a layer of loose connective tissue, and once again the same. Yes, once again the same. Another case, a related question. Now development. Yes, development of bones, ossification or osteogenesis. But we remember we need to differentiate between direct and indirect ossification. This is direct ossification, meaning directly from mesenchyme. How to identify? Yes, how to explain? I can identify this image as direct ossification directly from mesenchyme because here, first of all, I can see embryonic mesenchyme, yes, mesenchyme-like tissue. Just to, not to forget from where it is taken. On the example of low jaw, so this is flat spongy bone. You can say in general this is flat spongy bone which develop by means of direct ossification. My interest is this bright oxyphilic network of primary bone trabecula. Yes, primary bone trabecula, which are composed of what? Osteoid. So here I can see trabeculae of osteoid. Osteoid is a non-mineralized collagen matrix. We remember collagen shows oxyphilic properties. So these trabecula show strong oxyphilic properties, bright pink. Yes, bright pink color surrounded by embryonic mesenchyme. This is primary bone, yes, not organized. And in high magnification, then I can show you high magnification of the same trabecula, and then you can complete your description. Each trabecula, yes, is covered by what? By a single cell layer of osteoblasts. Each trabecula, yes, is covered by bone-forming cells, bone-producing, yes, osteoid-producing cells, osteoblasts. It would be perfect if you say they are columnar basophilic cells arranged as a single layer. You remember this? Yes, we discussed. And inside, I can see differentiating osteocytes. Yes, osteocytes, no cells. Sometimes you may also add sometimes osteoclasts may be visible. Multinucleated cells visible as clumps on the surface of bone trabecula. This is quite enough. Yes, you see, only general, only basic material. which I hope you remember. Yes, because we discussed in detail. Let's watch another case. Now, yes, now this is indirect. On the example of long tubular bone, what is the possible explanation? Why can I identify this, recognize this as indirect? What does it mean indirect? By replacement of pre-existing cartilage model. So if I can see at once within a single slide cartilage in conjunction with the developing primary bone, this is indirect ossification. Yes, this is the on the example of what? On the example of long tubular bone. It would be perfect if you remember the 5 zones of cartilage: resting cartilage, proliferating, hypertrophic cartilage, mineralized. Yes, you can also, if you remember this, it would be excellent. But and this is ossification line. Yes, the margin. Yes, the margin, the border of ossification. And I can say here I can see gradual replacement of pre-existing cartilage model by newly formed primary bone. Yes, primary bone. This is the so-called endochondral ossification, because inside this model I can see marrow cavities. Yes, developing marrow cavity. On the cavities on the periphery there is a perichondral bone. Collar. Yes, collar. Generally, this is enough for explanation. Yes, for confirming your decision. And this is again another case, just another variant of indirect ossification. Cartilage replaced by primary bone and ossification line between them. Once again, this is cartilage. Yes, hyaline cartilage, isogenous groups, mature cartilage group, zone. This is again endochondral ossification. Cartilage, and primary bone. This is direct ossification. Yes, direct ossification. Bone trabecula surrounded by mesenchyme. What is this? Let's look at these long striated, right? I can see striations.
Striated muscle.
Striated, but what variant of striated? I know 2 types of striated muscles, skeletal and cardiac. So this is
Skeletal.
Yes, this is skeletal. Taken from the, so this is skeletal, striated, somatic,
Involuntary. Involuntary.
muscles. Yes, taken from on the example of the tongue. Yes, tongue. Your explanation here longitudinally, I can see a structural and functional unit of skeletal muscles, which that is a muscle fiber. You should begin your description with a muscle fiber as a structural and functional unit. What is this, a muscle fiber? This is very long, cylindrical, unbranched, multinucleated structure.
With
Yes?
striations.
Showing striations, so bending. I can see dark and light bands. Nuclei are located on the periphery. Yes, multiple. So the principal formulation is long, cylindrical, multinucleated, unbranched.
Showing striations.
Yes, showing structure, muscle fiber, showing cross striations.
Nucleus periphery.
Alternation of light and dark bands with peripheral nuclei. Another case. Also striated, right?
Striated.
But now this is cardiac, yes, cardiac striated muscles. Why?
Intercalated disc.
First of all, you start with structural and functional unit. Here I can clearly define a cell, cardiac myocyte, as a unit,
Yes.
Structural and functional.
which spans from one intercalated disc, yes, one intercalated disc, to another intercalated disc. These cells are also elongated, prismatic, but mononucleated, sometimes binucleated. Then what is another difference? They are branched.
Branched.
I
Nucleus
can
at
see
periphery.
branches, or sometimes called lateral anastomosis. Yes, by these branches, cells can communicate with each other by lateral anastomosis. Intercalated discs are clearly visible. Nucleus, not very well visible here, but it is located centrally. Yes, and not peripheral. That is, generally, this is all that you need. So now a structural functional unit spanning from one intercalated disc to another intercalated disc, a cardiac myocyte, which is branched, mono or binucleated, organized, we can say, in three-dimensional network with centrally located nucleus. Quite enough. A couple of sentences. And this is one more case of muscles. Smooth muscles. Primitive description.
Smooth myocyte.
Yes, a structural and functional unit. If I show you in high magnification, I can define a myocyte,
Smooth miles.
which is mononucleated cell, yes, as a structural and functional unit, which is very elongated spindle cell with spindle shape, generally spindle-like morphology.
And
Centrally located elongated nucleus without striations, so smooth appearance.
Yes.
And oxyphilic properties. Yes, the cytoplasm is stained with eosine in pink. Yes. Well defined oxyphilic properties. Quite enough. Nothing else you can derive from hematoxylin eosine stain.
Yes.
This is our
Neurons.
neurons. Our last topic. Neurons, so nerve cells, taken from where? From the ventral horns of the gray matter of the spinal cord. Visualized by Nissl stain or toluidine blue. Here it's better to start with your explanation with specific shape. Yes, here I can see the cells with rounded cell body, large euchromatic nucleus, spherical nucleus, cell body of pericardion, and extending processes.
Dendrites.
Then, due to Nissl stain or toluidine blue, I can observe here chromatophilic substance or
Tigroid.
Nissl bodies or tigroid, totally filling the pericardium, and also including what? The initial portions of
Dendrite.
dendrites. Axons remain invisible. Yes, invisible in this case. In between them, I can also notice nuclei of protoplasmic astrocytes, because the location is gray matter. Yes, gray matter of the spinal cord, which contains protoplasmic type of astrocytes. Generally, that is all that you need. You may also say Nissl stain shows blue color. Yes, blue. This is toluidine blue.
Which
This
stain is
basic,
used?
Yes, Nissl toluidine blue or Nissl
And
stain.
for
Nissl
axon?
stain. For what? For
Axon.
not for axon. For cell bodies, for dendrites. Yes, which contain rough endoplasmic reticulum. So here we visualize rough endoplasmic reticulum aggregates, which are abundant.
Sister.
You may also say euchromatic nucleus. Yes, it would be perfect.
Mhm.
Euchromatic nucleus and nucleolus reflecting protein synthetic activity. These are myelinated fibers. Yes, myelinated. I will include only myelinated fibers because unmyelinated, not well distinguishable from sometimes tendon. So only myelinated fibers will be included. Here it's better to start with a specific stain. So these are myelinated fibers taken from peripheral nerve, split peripheral nerve. Yes, and you explain why we use osmium, as it is this is a stain with affinity for phospholipids. Yes, phospholipids.
Rich.
So it is quite reasonable to use this stain to visualize, to show, or to discover myelin sheath. Then what else? I'll take another picture. You can also complete your answer.
Neurolemocyte.
Yes, here I can see, for example,
Node of Renvia.
two internodal segments.
Internodal
Yes,
segment.
internodal segments separated by
Node of Renvia.
Nodal.
Nodal gap.
I can also notice sometimes neuralemma. Yes, this transparent.
Outermost layer.
Transparent layer of the Schwann cell cytoplasm. Sometimes I can find myelin clefts. Yes,
Schmidt-Lantermann
as
cleft.
small spaces inside the myelin layer. Inside, I understand that
Schmidt-Lantermann.
inside there is axial cylinder, an axoplasm or axon cylinder, but it remains invisible.
Yes.
Unstained in osmium. This is quite enough, yes, for explanation. Once again, you see, I am based on the legend. Yes, all the definitions that are shown in legend, but sometimes students just an example of incorrect answer. Myelin, clefts. So I identify this slide because I can see clefts, nodes of Ranvier, internodal segments. No, you need to make a story, a description. Just listing definitions is not enough.
Yes.
This is not a description. Make 3-4 sentences. This is should be like a short story, a short description. This is again
Neuron.
Yes, neurons. And one more portion.
Blood
of
cells.
Blood cells. Yes, blood cells will be included. Today I will not spend time repeating once again these images, but I will put it in Moodle. They will be directly the same. You remember we exercised few weeks ago. You should be based on the relative size. You start with the relative size in comparison with erythrocytes. Larger than erythrocytes, smaller or similar in size. Then you describe the shape of the nucleus, the density of the nucleus, condensed or
Diameter.
or probably weakly stained, darkly stained nucleus, euchromatic, heterochromatic, then segmentation of the nucleus, segmented or probably
Granular
unsegmented
structure.
mononuclear. Then staining properties of the cytoplasm, granules, are they present?
Ultrastructure.
Cytoplasm, basophilic, weakly basophilic, weakly oxyphilic. So today we will not stop once again on it. And some more slides. Now from the beginning. Now epithelium. So let's try to identify what is the type of epithelium.
Columnar.
Ciliated.
Ciliated.
Ciliated. So it is, first of all, it is
Cuboid.
simple.
Simple.
Single layer, columnar. This is low magnification. This is high magnification. Yes, over portion. Here I can see from where? Here I can see obviously two villi, intestinal villi. Each villus is covered by intestinal villus. Yes, it's covered by simple single layer columnar epithelium, and to give full classification, this is simple single layer columnar brush bordered epithelium. How can I identify? On the surface, on the apical surface, I can see a brush border. Yes, a brush border. This epithelium consists of, you remember, two major types of cells. Absorptive enterocytes. Enterocytes are absorptive cells. These
Goblets.
with basally located nucleus and apically brush border, and another type
Goblet
cells. Yes, goblet cells.
Fat
They are
storing.
less numerous. And
Fat storing.
all cells attached to basal lamina underlined by lamina propria. This will be quite enough for morphological description. This is just to not to forget about blood cells. Yes, this is neutrophil, segmented polymorphic,
Multilobular.
mature neutrophil.
Mature neutrophil.
This is again
Loose, fibrous
fibrous. This is again
Cardiac muscle.
skeletal
Skeletal.
striated
Skeletal.
muscles. These are
Cardiac muscle.
Yes, here we clearly see branching. Yes, branching, intercalated discs. So this is one cardiac myocyte. Yes, spanning from one disc to another disc, and the nucleus, single nucleus. And here we clearly see that they are arranged as a three-dimensional network.
Prismatic.
Yes, bilateral anastomosis, prismatic shape. Yes, prismatic.
Lateral
It is
anastomy.
prismatic shape. Yes, prismatic. What is this?
Neuron.
Once again, what can I see? Many
Neurons.
canaliculi. Yes, and lacunae. Yes,
Bone.
where osteocytes may be present. Yes, this is secondary lamellar bone, but in high magnification. I can't see here osteons. This is just a single osteon. But I can see osteocytes, elongated almond-shaped cells, yes, with occupying lacuna with canaliculi. This again, yes,
Simple epithelium.
small intestine.
Intestinal villus.
Don't forget to give full classification.
Brush
Simple,
border.
yes, simple single
Simple
layer,
single
columnar,
layer.
brush
Columnar
border.
brush border.
What is this? This is, let's first watch low magnification, total view. This is the first topic.
Glandular.
Yes, glandular epithelium, and we watched only one example of glandular epithelium. So this is, if you remember, this is sebaceous gland.
Sebaceous. Sweat glands.
How can you explain why glandular? First of all, here I can see something arranged as lobules. Yes, lobular structure. If I can see lobular structure, several branched lobules. If these are lobules, any case, this is a gland. Now let's several lobules or secretory portions. Yes, secretory portions converge into a single duct, short duct, which opens or empties into what? Into hair follicle. Yes, hair follicle. And we remember that only sebaceous glands are associated with hairs. Then we need to we need to give full classification, structural classification, morphological classification of a gland. You should say, this is, first of all, an exocrine gland. By morphology, this is branched, but simple, acinar gland with holocrine type of secretion.
Holocrine.
So it is branched because I can see several lobules. Yes, several lobules, several terminal acini with an acinus as a structural unit. It is simple because here I can see only one duct. It is simple. And by the type of secretion, it is holocrine, which means that it undergoes total destruction upon secretion. And in high magnification, you can also, it would be perfect if you say, yes, the major differentiated cell type is a sebocyte. Yes, sebocyte, large rounded cell, glandular cell, which accumulates lipid droplets. These droplets fill the whole cytoplasm, and upon terminal differentiation, they undergo total destruction closer to the duct and disintegrate, totally disintegrate. That is all for sebaceous gland. So lobular structure. Yes, if you can see lobules, this is a gland. Exocrine gland associated with hair follicles. What is this?
Stratified.
Ciliated.
Pseudo-stratified.
Columnar epithelium taken from respiratory tract. Yes, another name for pseudostratified ciliated epithelium is respiratory epithelium, taken from trachea. You need to explain why pseudostratified. What does it mean?
Single layer, but they are single layer.
Yes. I can count. If this is basal lamina, I can count 1, 2, 3, at least 3 levels of nuclei, not layers, levels of nuclei, due to difference in cell sizes. Yes, cells have different sizes, different probably specifications. So here I can see false stratification, false appearance of being stratified. And not forgetting about cilia. Yes, this is a layer of cilia, not brush border, cilia. What is this? Mesothelium. Yes, mesothelium. Once again, how to explain why mesothelium? So yes. So first of all, the major, if you remember, the major morphological feature of any epithelium, continuous layer. If you observe something that grows as the continuous layer with no spaces between the cells, no extracellular matrix, this is the major morphological distinguishing feature of epithelium. So cells are tightly bound to each other with zipper-like. Yes, zipper-like junctions, we can say, or zigzag-like, yes, borders, with no spaces, with no extracellular matrix. And another distinguishing feature is the regular shape. And these cells have generally polygonals. This is mesothelium, full classification, simple squamous epithelium. But this is superficial view. Yes, we look at the cells superficially. Superficially, they are polygonal. A typical shape for squamous, simple squamous epithelium. So the primary property is a continuous layer with no spaces between the cells. This is again a respiratory pseudostratified epithelium. Yes, pseudostratification. 1, 2, I can count even 4, sometimes 3, 4 levels of nuclei. What is this? Not transitional, stratified, but what? Stratified, squamous, keratinized epithelium or epidermis from the thick skin. Your explanation. Here I can observe at least 4 or 5 layers of cells. It would be perfect if you remember the names of this stratum basalis, stratum spinosum, granulosum, lucidum, and corneum. it would be excellent. But what else you need to complete? So first of all, 4 or 5 layers of cells, well separated, with the thickest superficial corneal level layer, cornified, consisting of corneocytes, yes, cornified cells, and another the most distinguishable layer number 3,
Granular.
granular layer,
Granular.
yes, dark violet, dark basophilic accumulating keratohyalin granules, yes, basophilic granules. This will be enough for description, for identification. And one more interesting type of epithelium. Now we remember that how to identify. We remember that epithelia are stratified epithelia are classified by the shape of only superficial cells.
Yes.
There is one interesting case where superficial cells are the largest. Urothelium. Transitional epithelium, where superficial cells are represented by umbrella cells, umbrella, the largest cells. This is our case. Yes, superficial cells, the largest with dome-shaped apical surface, dome-shaped. Generally, this is enough. You should also say this is a specific type of stratified epithelia, transitional. It would be perfect if you can explain what does it mean, transition. Yes, it has no constant shape, no constant number of layers. It transits from different shapes, different layers, depending on the stretching degree of an organ distension. On the example of urothelium of the urinary bladder, this will be enough. And the last image, the last slide. The last type of epithelium, also stratified. Yes, I can see several layers of cells, so it is stratified. But we need to watch the superficial cell layer. So it is squamous. Yes, superficial cells are flattened, but nuclei preserved. So cells remain alive. That is why it is non-keratinized. If you remember the three layers: basal, spinous, and squamous superficial cells, yes, it is perfect. And your explanation is that I can classify this epithelium as squamous non-keratinized due to the shape of superficial cells. They remain alive, they preserve nuclei, they are flattened, so this is non-keratinized, which is in contrast to this case, yes? Superficial layer cornified, dead, totally dead cells, corneocyte. That is all. You see just general description, yes, general material with no specific information about functions, mechanisms, no. Only what is visible. Your short description should include only visible morphological structures. Prepare in advance 3, 4, 5 sentences of description, of explanation. Today or probably tomorrow I will load these images, all these images, directly the same in Moodle. And you prepare hematopoiesis. Next Thursday, first within 15 minutes we discuss this topic, then I will test everyone. For